Hi everybody, Miss Calabrese here. So in this video, we're going to be talking about the processes of cell division. So how eukaryotic cells copy themselves and separate. So two separate processes here, mitosis and meiosis. All right, so mitosis is also known as somatic cell division. So this means body cells. So it's basically every cell in your body except for the eggs and the sperm cells. All right, so in order to understand mitosis, we kind of have to put it in the context of the entire cell cycle. So a cell cycle is basically the life cycle of a cell. And it consists of two main parts, and those are interphase and mitosis. So interphase is everything that happens between rounds of mitosis. All right, so here's the entire cell cycle, and we'll start here at the end of mitosis and then kind of work our way around. So starting here, the first phase that we get to is this G1 phase. So this is our first growth phase of the cell. So right after a cell is born, it's going to start getting to work. It's going to be very metabolically active. That means it's going to be doing a lot um, of the transcription and translation, a lot of that protein synthesis or gene expression that we talked about last time. So we're making proteins, we're making enzymes, we're doing the work of that cell. Now, the work of that cell is going to vary depending on what type of cell it is, but this is the cell doing its normal job, right? The phase after G1 is called S phase. S stands for synthesis here. In the synthesis phase, that's where we're going to replicate the DNA. So this is where DNA replication happens. So we take our one strand of DNA or our single DNA, and then we make an identical copy of that DNA. After synthesis, then we go into G2 or growth 2. So cells continuing to grow, we're still synthesizing proteins, and we're doing one more thing where we are replicating our centrosomes. So this is the pair of centrioles that we talked about in the cellular anatomy video, right? So these three phases, G1, S, and G2, all together are known as interphase. Right? So this is what's happening when the cell is kind of doing its normal job. After G2, then we start mitosis. And mitosis, we can divide into four basic steps. And those are prophase, metaphase, anaphase, and telophase. Those are the four steps of mitosis. And we'll talk about what's happening in those four steps. And then at the end of mitosis, we go from having one cell to having two separate cells that are identical. And those two cells will now start the process all over again. Right, so these four steps of mitosis, PMAT, prophase, metaphase, anaphase, and telophase. What's happening in each of these steps, I'm kind of go through them one by one. In prophase, a lot of stuff is happening. One, the chromatin, which is that combination of DNA and histone protein that you have in your nucleus, is going to start to condense into chromosomes or chromatids. All right, so we're condensing that DNA and kind of packaging it up like we're getting ready to move. So packaging it all up nice and tight. We're going to start to dissolve the nuclear envelope. So it's going to start to break down. So the chromosomes are going to become more visible. The nuclear envelope begins to break down. The nucleolus is going to disappear as well. And so then we're going to move into metaphase. So in metaphase, the sister chromatids, so these identical arms of the chromosome, are going to line up in the middle of the cell. So think M stands for middle. This is where chromosomes line up in the middle of the cell. In anaphase, this is when they start to pull apart from one another. So think A anaphase, away or apart. So identical chromatids are going to be pulled to opposite sides of the cell. And the structures that are pulling them to opposite sides are those centrioles. So the centromere is the name for the pair of centrioles. It's going to pull it to opposite sides of the cell. And then the last phase here, telophase, when you think telophase, think T for two. So this is where we're going to reform two new nuclear envelopes around those two identical sets of DNA. So at that point, at the end of telophase, we've now got two new nuclei. Right, so and that's all mitosis technically is nuclear division, where we make a copy of the nucleus, and now we have two nuclei. Right, so here's a look at what that is visually. So prophase here, the first step, we can see that the chromosomes and the nucleus are condensing. The nuclear envelope is beginning to break down. I want to point out your centromeres here. There's one, and there's one. These that were duplicated at the end of G2 are now starting to migrate apart from one another. So they're moving to opposite ends of the cell. So I'm going to skip prometaphase and go right to metaphase so we can see here's a centromere on one side, here's a centromere on the other side of the cell. So these have migrated to opposite poles, and then behind them, they're dragging these lines here. These lines represent spindle fibers, which are basically microtubules that are coming from those centrioles and attaching to the chromosomes that are now lined up directly down the middle, the equator of the cell. All right. Next step is anaphase, and that's when these sister chromatids are going to be pulled apart, so they start migrating to opposite ends of the cell. And then telophase, we've got two new nuclear envelopes that are forming around those now separated chromosomes. Right, and at the end of telophase, that's the end of mitosis. Cells aren't separated yet, but the nuclei are. Right, the last step of the process is cytokinesis. And cytokinesis is when we actually separate the cells. So we're going to pinch those two nuclear membranes apart from one another. All right, so yeah, cytokinesis is the division of the cytoplasm. Right, so it's a separate process from mitosis. It does start in late anaphase, but it completes itself after telophase. So what'll happen is you start by producing what's known as a cleavage furrow, which is like this indentation in between the cells that will eventually be separated. And then that indentation kind of just gets smaller and smaller and smaller, pinching off those two cells so that we have now two daughter cells that were identical to the original cell. All right, so here's another look at it where you can see the drawings and then also some actual micrographs of what's happening here. So interphase is represented up top here, so we can see this is the cell just kind of doing its normal work. Everything that's involved in cellular synthetic processes is happening in interphase. Towards the end of interphase, then we will duplicate those centrioles, and then we will begin prophase. And in prophase, those centrioles are going to start moving farther apart. Nuclear envelope is going to break down. The chromosomes are condensed, and then we move into metaphase. In metaphase, the chromosomes are going to be lined up down the center axis. In anaphase, they're going to start to be pulled apart. In telophase, we're going to get two new nuclear envelopes, and then we'll undergo cytokinesis to officially split those into two identical daughter cells. Okay, so that mitosis is for somatic cells or body cells. Reproductive cell division is a different process entirely. So this reproductive cell division is called meiosis. And meiosis is how we produce egg and sperm. So this is the only cells that use meiosis are the egg cells and the sperm cells. So this is important because meiosis is going to be a very different process from mitosis. Some of the steps are the same, but our end result is very different. So in mitosis, remember we're producing two identical daughter cells. In meiosis, we're going to be producing non-identical daughter cells, and instead of two, we're going to make four of them from a single precursor cell, right? And each one of those daughter cells that ends up being either an egg or a sperm is going to be what we known as what we call haploid. So haploid means it's got half the number of chromosomes as was in the original cell. So for example, your somatic cells, the cells of your entire body, those are diploid, meaning they've got you've got pairs of chromosomes, one set from your mom and one set from your dad. So in diploid cells have 46 total for 23 homologous pairs. Homologous means they correlate with one another. But when we make these egg and sperm, we want to cut the number of chromosomes in half because we want 23 in an egg and 23 in a sperm. So when they meet, we total up to 46 again. Okay. So as far as cell reproduction goes, all of your cells are going to have the identical DNA, no matter what type of cell we're talking about, well, except for egg and sperm. We're all going to have the exact identical DNA. And remember, we talked last week about this process of differentiation, how we go from being kind of like a stem cell that's capable of becoming multiple different cell types into becoming all of those different cell types. You have roughly 300 different types of cells in your body. So differentiation occurs by turning on and turning off certain genes. And that's what's going to make a skin cell different from a muscle cell. So we have uh, proteins called transcription factors. And these transcription factors are either going to promote transcription of certain genes or inhibit transcription of certain genes. So basically turning genes on and off that are going to lead us towards differentiating into a certain type of cell. Okay, a little bit of terminology associated with cell reproduction. So hypertrophy is increasing the size of an individual cell. So hyper means more, troph refers to growth. So basically overgrowth of a cell is just getting a little too big. Atrophy is when a cell is going to decrease in size. So it gets a little bit smaller, it starts to shrink. So hypertrophy, overgrowth, atrophy, shrinking. Hyperplasia is an increase in cell reproduction. So instead of cells getting bigger, they're just reproducing more. And anaplasia is the production of abnormal, undifferentiated cells. So basically producing immature cells that don't differentiate properly. Okay. Neoplasms or tumors, these are abnormal growths of cells. And basically this is what happens when mitosis is not well controlled, right? So normally your cells undergo mitosis on a set time schedule. There are checkpoints that the cell has to kind of follow along in order to know when it's going to undergo mitosis. If a cell is damaged in some way and it kind of blows past its own checkpoints, then we can get neoplastic growth. And so that's when we begin to form tumors. And tumors can be classified as either benign or malignant. So benign tumors are usually encapsulated, they grow slowly, and they don't cause too much trouble. Malignant tumors are not well encapsulated. They grow very fast and they're undifferentiated cells, so very immature cells that are just kind of running wild. Malignant tumors are capable of metastasis, which is when cells that are going to leave that primary tumor and move to a new location. All right, so things that cause cancer, so things that can cause your cells to undergo uncontrolled mitosis. So there are genetic factors, certain oncogenes or cancer genes that make you more likely to develop certain types of cancers. Carcinogens are chemicals that can alter the genetic activity, so basically cause mutations in the cell. Age can contribute to the likelihood of developing cancer because as you age, your cells are just not going to be functioning as properly anymore. Environment, so exposure to things like radiation and UV light, and then some viruses. So viruses, because of the nature of their reproductive patterns, have to get inside the nucleus of your cell and mess with your DNA. So some viruses do cause cancer. Okay, so as far as treatments of cancer, obviously it's going to be different depending on what types of cancer we're talking about, but surgery to remove the abnormal cells, chemical therapy or radiation therapy to try to destroy those undifferentiated cells. There's a lot of new immunotherapy techniques that are happening, which is basically boosting your immune system to attack its own cancer cells, which is very promising research. So I hope this was helpful. Let me know if you have any questions.